Good day, everyone, and welcome to the laboratory orientation of the accessory organs of digestion. The following organs are the areas of the or are the areas to be studied for this orientation. We have the liver, the gallbladder, and the pancreas. Okay, so let's start with the liver. The liver is known to be the largest internal organ of our body, and the main function of the liver is production of bile. Okay, one of the more important functions of the liver is bile secretion. As you can see in this slide, the liver parenchyma, which are known as your hepatocytes, is organized as thousands of small hepatic globules, in which the liver cells form hundreds of irregular plates that are arranged radially around a small central vein that is in yellow circle. Okay. The hepatocyte plates are supported by a delicate stroma of reticulin fibers, and peripherally, each lobule has 3 to 6 portal areas with more fibrous connective tissue. Portal triad in the portal area will be discussed in the next slides. As you can see, this is the hepatic globule or the classic globule. They are described as hexagonal in shape, no? As you can see in this illustration. They are in central vein in the center, at the center, with 6 portal veins or portal areas or portal triads on the sides. No, on the left side of the screen, you can see that at the center of this hepatic globule is the central vein with 6 portal area or portal triad. No, on the right side of the screen, as you can see, this is the central vein, and there are 1, 2, 3, 4, 5, and 6 portal area or portal triad on the sides. Okay. As mentioned, the liver's parenchyma is the hepatocytes, or known as your liver cells. Other characteristic of the hepatocytes, if we ask you what is the shape of the parenchyma or what is the shape of the cell, it is polygonal or polyhedral in shape. The number of nucleus of the hepatocytes is, take note, it's either mononucleated or binucleated. You have to answer mononucleated or binucleated. If asked what is the location of the nucleus, they are centrally located, and the arrangement of the parenchyma, which is the hepatocyte, they are arranged in rows, cords, or columns. Okay. Now, next slide would be hepatic sinusoids. Hepatic sinusoids are actually found in between the hepatocytes. They have a unique microvasculature that serves as the principal site of exchange between blood and the perisinusoidal space, which is the space of this, specifically found in the liver. On the left side of the screen, as you can see, the blue arrows, they are your hepatic sinusoids. And on the left side of the screen, as you can see here, found in between the hepatocytes are all your hepatic sinusoids. Okay. All right. Now, as mentioned a while ago, the hepatocytes can be seen, or the number of nucleus is either mononucleated here or binucleated. Okay. This is also binucleated. So the hepatocytes they have this mononucleated or binucleated. Now, the cell that is pointed right now is actually a von Kupffer cell, specifically found in the liver. Okay, they are your macrophages found in the liver. They are stellate in shape, macrophage. They are found within the sinusoidal lining. One of the functions of your von Kupffer cell, or the cell that is pointed right now, is they detect and phagocytize effete erythrocytes. Also, another function of your von Kupffer cell is they act as antigen-presenting cells. They remove bacteria and debris in the portal blood circulation. Okay. So there you have it. There you have the hepatocytes, and the cell that's pointed right now in the blue with the black arrow is your von Kupffer cell, which serves as your macrophage or antigen presenting cells. Okay, next slide. Okay, the portal triad is actually found in the portal area, and there are three structures that are found in this portal area, namely the portal vein, the hepatic artery, and the bile duct. So let's differentiate one from the other. The portal vein is actually a venule branch of the portal vein with blood rich in nutrients but low in oxygen. The hepatic artery, on the other hand, is an arterial branch of the hepatic artery that supplies most of the oxygen. And lastly, the bile duct, it's actually one or two small bile ductules of cuboidal epithelium branching on the bile conducting system. Okay, next slide would be another picture of the portal triad. No, obviously this is a portal vein, as described before in histology of the blood vessels. Portal vein has a collapse or larger lumen. Look at the hepatic artery, compare it to your smaller hepatic artery, a smaller lumen of the hepatic artery. Also, one differentiating factors of vein and on artery is that, look at the hepatic artery, it has a thicker tunica media. All right, this is a branch of bile duct, or simply, if we ask you in the examination, identify specific structure pointed found in the portal area, you may answer bile duct. What are your clues? Now, what's the clue that this is a branch of a bile duct or simply bile duct? Look at the lining epithelium. No? It forms a single layer of cuboidal cells. Thus, the lining epithelium of the bile duct is simple cuboidal epithelium. Okay, again, these are the components of your portal triad. You have the portal vein, the hepatic artery, and your bile duct. Your clue in your bile duct is that it is lined with simple cuboidal epithelium. Okay, discussed to you in the lecture, we have the different lobules or shapes that found in the liver. The hepatic lobule obviously is hexagonal. As described before, it has central vein at the center with six portal triad at the sides. The portal lobule is described as, look at the green triangle. It's a triangular in shape. Portal lobule, your clue here is that at the center would be the portal area or the portal triad with three central veins on the sides. Okay, so that would be the portal lobule. The other one is the hepatic acinus. When you say a hepatic acinus, it is shaped like a diamond. So it has four sides. It is composed of your two central veins, and you have your two hepatic or two portal areas or two portal triads. As you can see in this slide, the one that is pointed right now is the central vein, and this is the portal area with portal triad. And in the liver acinus, we can actually distinguish it with three zones. Okay, the blue one, the yellow one, and the green one. Zone 1 would be the most oxygenated area or zones, primarily because they are found near, no? They are the nearest to the portal area. Next would be the zone 2, the yellow shaded area, and the zone 3 would be the green shaded area. The zone 3 class is the least oxygenated, primarily because they are found farthest from the portal area or the portal triad. Most commonly asked questions in the examination: which of the following zones is the most susceptible zone or area comes when there is a liver injury? Then the answer would be the green shaded zone or the zone 3. It's because they are found the farther from the portal area. Okay. All right, so that concludes the liver histology. Next accessory organ of digestion would be the gallbladder. As described, they are hollow, pear-shaped organ, and it contains 30 to 50 ml of bile. Now, the main function of your gallbladder is to store the bile that is produced by the liver, and it also concentrates bile. Okay, that is the function of your gallbladder. And the gallbladder is composed of the following layers. First, we have the mucosa, and it has lining epithelium and lamina propria. This one is your tunica muscularis. Next area or layer would be the tunica serosa, and the one that is adherent to the liver is your tunica adventitia. In the not the layer, but this is actually part of the tunica serosa. That is your mesothelium. Okay, the gallbladder. Okay, its mucosa, its lining epithelium is described as simple columnar epithelium. Okay, it also has this sublayer of mucosa, which is your lamina propria. And the characteristic feature of your gallbladder is found in the lamina propria. And these are your Rokitansky-Ashkoff sinus, or simply RA sinus. Okay? All right, so that would be the most important things that you need to remember in the gallbladder: its function, its layers, its lining epithelium, and the one that is encircled in red circle would be the Rokitansky-Ashkoff sinus, or the RA sinus. That is your characteristic feature of your gallbladder. And let's go now to the last accessory organs of digestion, which is your pancreas. The pancreas produces 1.5 liters of alkaline juice per day. No, that is directly drained into the duodenum, and that would be the exocrine part. The endocrine part is composed of your islets of Langerhans, or your pancreatic islet. They produce hormones. Okay. Now, the pancreas, no, as you can see here in this slide, they are composed of, as mentioned, these are the lighter in staining islets of Langerhans. That is the endocrine part. And the one that is with yellow arrows are actually our pancreatic ACINAR cells. And that would be your exocrine part. The islets of Langerhans, as you can see in this slide, is the endocrine part. They secrete hormones such as your insulin and glucagon that control your blood sugar levels throughout the day. Insulin, as you all know, is being secreted during fed state or after eating, while your glucagon is secreted during fasting state. As glucagon increases the blood sugar, your insulin now decreases your blood sugar level. Okay. Shown to you in this slide are your pancreatic ACINAR cells, the one that is in blue circle. And at the right side of the screen are your central ACINAR cells. Obviously, they are found at the center of your pancreatic ACINAR cells. Another picture of your pancreatic ACINAR cells, no? This is your central ACINAR cells that are found at the center, and the one that is found at the periphery are your ACINAR cells. And the whole area right here is your pancreatic ACINAR cells, which is your exocrine portion of your pancreas. The pancreatic or alkaline juice that is produced here by the ACINAR cells they will directly drain into the duodenum in case of a fatty diet or an alcohol ingestion. Okay, and that concludes the laboratory orientation of your accessory organs of digestion, which are your liver, your gallbladder, and your pancreas. Thank you for listening.